Thank you for a great introduction. Like I said, I'm going to ignore all of the comments about Tasmania. It's a great place. And so I'm going to bring this session to almost a full circle and tell you again another little epigenetic story that we have been working on. As Terry closed his talk, he mentioned some centres of excellence in epigenetic research, and I've been fortunate to work at two of those, the University of Southern California and now at the Garvan Institute. And this is a collaborative story. I started this work when I was in Peter's lab there, and now with the support of Sue and my colleagues here in this lab, we've been able to bring this story to a little bit of a close. So, the title of my talk is Massive Reorganization of Nucleosome Distribution and DNA Methylation at Distal Regulatory Elements in Cancer. I won't be able to show you all of the data today, but hopefully I'll give you some of the take-home messages and the exciting findings that we've been able to find in this story. So I thought I'd start right back at the very beginning. We've had a great introduction to epigenetics today, but I thought I'd start with this because it really highlights the importance of epigenetics in that if we have our DNA sequence, which is the same in every single one of our cell types arguably, epi literally means the modifications above this DNA sequence. And these can be repressive modifications which turn genes off, or active modifications which allow genes to be turned on in the right context. So we can think of this as genetic punctuation. If we have all of the same letters outlaid here on the left, when we put the punctuation in, finally that story makes a bit of sense and we get full stops so that we can take a breather and we can read the sentences that are meant to be read. So in a more complicated manner, we've seen some of these diagrams already today. We see that the epigenome, or the genome, is not linear. We have the same 2 meters of DNA in every single cell of our body, and this must be wrapped and compacted so that it can fit inside every cell. But it has to be done in a very precise way so that the right genes are expressed at the right time. And this involves and relies very heavily on epigenetic mechanisms. And we've already heard a lot today about DNA methylation from Terry and histone modifications from Suda. And I'm going to add in another little bit of a story, and Suda did mention this, and that's nucleosomes. Without the nucleosomes, we couldn't wrap the DNA around the histone proteins, which form the nucleosome and allow this DNA to be compacted. We wouldn't have histone modifications, which occur on the tails of the histone proteins. And we have some evidence to show that the nucleosome is critical for DNA methylation to occur. So I like to think of this as the center of the epigenome, but also for another reason in that, as I mentioned before, we have to have regions of open chromatin so that we can have expression occurring at the right time, and these are called nucleosome depleted regions. And I'll come back to this during my talk. So I'd like to start by highlighting the fact that there's been a lot of interest in epigenetic mechanisms and how these go wrong in cancer. And that forms the basis of the model that I'm going to talk to you today about. And what we do know is that promoter regions have been quite extensively epigenetically studied, and we know that they become epigenetically reprogrammed in cancer. So if we take a normal cell and we look at this schematic diagram, we have genes that are normally expressed. They have nucleosome depleted regions to allow the transcriptional machinery to bind, and we have the active epigenetic marks associated with these regions, allowing these genes to be expressed. In the cancer cells, we see some dramatic changes in the epigenome. We see the output is gene silencing in many cases, and this is the result of abnormal DNA methylation. I'd like to also highlight that we lose the nucleosome depleted region, and we also gain repressive histone modifications at the same time. Obviously, there's a whole host of different ways this can occur. This is the most commonly studied pathway from expression to silencing. We can also see from repression where we get an exchange of histone marks from a repressive state to a more compact epigenetic silent state by the gain of DNA methylation, and of course some more complicated stories in the middle. We can also get the reverse. So in addition to abnormal silencing of genes due to epigenetic changes, we can see the abnormal activation of genes, such as this gene here that was normally silenced by DNA methylation, but becomes abnormally expressed in cancer cells. And we could consider these things like oncogenes, for instance. So the question was, after we know all of this can occur at promoters, but the one thing that I was really interested in asking was whether distal regulatory elements such as enhancers, which we've heard a little bit about this morning, are also subject to epigenetic silencing in cancer or become reprogrammed abnormally. And to answer this question, I've focused on DNA methylation and nucleosome positioning. And I will show you a new technique that we developed to be able to answer this question. But I'd just like to point out that we had some preliminary evidence that this actually does take place. Using the MyD1 gene, I compared the epigenetic signature of MyD1 promoter and enhancer in cells in which it is active and expressed. And I compared the epigenetic signature in cells, in cancer cells, whereby it's not expressed and we know that the promoter is heavily methylated. And then we studied what happens at the enhancer. And so what we can see is, using MyD1 as an example, we were able to show that, in fact, the MyD1 enhancer also becomes DNA methylated, similarly to what we saw at the promoter. And we also see this nucleosome popping right in here at the center of the enhancer, closing this nucleosome depleted region and blocking access to all of the other transcription factors and other proteins that might bind to allow this gene to normally be expressed. So these were the questions we wanted to ask. Are these epigenetic changes that we're seeing at MyoD common, and do they occur genome-wide? And likewise, are these epigenetic changes occurring in many different types of cancer, or are they limited to one type of cancer? So to answer that question, we devised an experimental model where we looked at normal cells and cancer cells. We used a model of prostate and breast cancer to investigate these questions, and we used human memory epithelial cells and prostate epithelial cells as our model of normal cells, and we used the MCF7 breast cancer cell line and also LNCAP and PC3 prostate cancer cell lines to answer this. And so what we wanted to know, to answer these questions, we used a combination of genome-wide techniques. We used ChIP-seq, and we looked at lots of different active and repressive marks. I'm not going to show you all of those today. And we also devised a technique called GNOME-seq, nucleosome occupancy and methylation sequencing, to investigate DNA methylation and nucleosome occupancy in single molecules. And we wanted to know, these comprehensive data sets would allow us to answer lots of different questions, but in particular, whether the silencing of enhancers is limited to changes in histone modifications, or whether it's a comprehensive change whereby we see also changes in DNA methylation and nucleosome positions in order to shut down these enhancers and change gene expression profiles in cancer. So how does nucleosome occupancy and methylation sequencing work? I've just done a little bit of a diagram here showing that if we treat our cells with an exogenous methyltransferase, I've entitled it GPC methyltransferase. It has an entirely different name if you want to order it from NEB, but nonetheless, we take advantage of the fact that normal mammalian cells, like our human cell lines that we're using here, do not exhibit methylation on the cytosine when it is preceded by a guanine. As Terry mentioned, normal endogenous methylation occurs in a CpG context. So if we treat cells with an enzyme that can methylate the cytosine in a GPC context, we also know that it only methylates those cytosines when there's regions of open chromatin, such as nucleosome depleted regions, or where there's tight binding transcription factors. So after we extract the DNA and we do the bisulfite conversion that Terry mentioned before, and we can either do genomic sequencing of individual molecules at a locus of interest, or we can do a genome-wide sequencing experiment, we can see where there were regions of open chromatin or nucleosome depletion, or regions where there were nucleosomes bound. So we adapted this technique, which was originally being performed on regions of interest, such as key enhancers or promoters, such as the MyoD example I showed you. And we were able to couple this with next generation sequencing, and through some very fancy bioinformatics, of which I did not do myself, but I can attribute to the work of Aaron Statham in Sue's lab. He was able to align all the data, count the changes in GPC methylation, and show us where there were changes in nucleosome occupancy in the normal and the cancer cells. So what can we see? Well, this is one of the very first main messages that we saw as a result of that analysis, and we can say that now that nucleosomes are organised throughout the genome. Originally, we were of the opinion, as were many others, that perhaps nucleosomes were only organised around key regulatory regions. And throughout the rest of the genome, in fact, there was some kind of abnormal distribution, and the nucleosomes were a little bit dynamic. But what this data shows here, if we look at the the green line here, we can see phasing of the nucleosomes indicated by these bumps here, and a very nice nucleosome depleted region at the centre of our region of interest. We can also show in the exact same molecule a dip in DNA methylation, which is consistent with the nucleosome depleted region. But what was interesting from this data was that we could see phasing even when there was no nucleosome depleted region present, which was quite surprising. And similarly to this, we saw an increase in methylation at these regions. And this was not seen when we looked at the same kind of data using DNase hypersensitive, which is another measure of nucleosome depletion. And we did not see it when we lose the binding of key transcription or regulatory factors such as CTCF. So to the main point of the talk, in that how do nucleosome positions change in cancer cells? What we did, I've put this diagram up here to show you again that we have promoters and enhancer regions, and we divided the genome by integrating our ChIP-seq data with the GNOME-seq data and asked where are the nucleosome depleted regions in the cell. And we can see here, I'm just going to point to the enhancers because they were my focus, but you can see in the normal cells we have a high number of NDRs overlapping enhancers, but in the cancer cell model we see a drop in the nucleosome depleted regions, suggesting that they're being shut down by the gain of nucleosomes at these regions, and we saw the same in the prostate model. This was just to highlight, if we look at the change in DNA methylation as well as the change in the change in histone modifications in nucleosomes, this is just reiterating the point that promoters are silenced by the acquisition of methylation and nucleosomes in the cancer cells. This was our proof of principle. As I said, this has been very well studied before. So promoters there. And if we look at enhancers, which are located distal from the promoters, we see the same thing can happen, that we gain DNA methylation at the same time that we gain nucleosomes. So everything's getting shut off at once. And if we zoom into the data, this is what it kind of looks like. If we divide our genome into different states, here we have two representative enhancers that become heterochromatinized in the cancer model if we look at those genomic regions. What was quite interesting was when we looked at another subset of enhancers that lose the NDR and gain DNA methylation, but don't exhibit a change in their histone modifications. And this was quite exciting because we don't see that at either the promoters or the other regulatory elements that we were looking at. So just to finish off, I haven't had time to show you a lot of the data that we generated, but to point out that we do see this large-scale change in methylation and nucleosome positions in the cancer cells. We were able to show that the nucleosome-depleted regions are key to DNA hypermethylation susceptibility, and this contributes to the epigenetic silencing of these distal regulatory elements in cancer. I haven't been able to show you all the data, but we looked at the transcription factor binding and how this can impact nucleosome organization in the cells, and we show that this can be critical. And so finally, just the main point is that in addition to promoters, there is a substantial disruption at distal regulatory elements in cancer. We see a change in chromatin architecture and histone marks that might provide another mechanism of epigenetic change in cancer. And with that, I'd like to acknowledge all of the people that have supported me through this, the team in the US at USC, and of course, my colleagues here at the Garvan Institute, and our funding bodies, and in particular the Cancer Institute. Thank you.
Thank you for that talk, Phillipa. It's another interesting view of the genome, definitely. Are there any questions from the audience for Phillipa? Surely somebody has got some question that they cannot resist to ask. Thanks, Amanda.
Great talk, Pip. As you know, I don't know a whole lot about epigenetic regulation, but do you think that that difference where you saw change in the nucleosome positioning but no changes in the histone, is that a general function of enhancers, or is that just something specific to the enhancers that's going on in cancers?
That's a good question. So we've only used cancer models, so I couldn't sort of extend that to other disease states. I think I mentioned that we did not see the same happening at promoters or other distal regulatory elements such as insulators. So we see a loss of the histone marks in addition to the change in DNA methylation and nucleosomes. Also, if you look back at the numbers, there's actually substantially less in this category than here. And I think we need to go and have a look closer at those subset of genes and see what's going on.
We have another question.
Great talk, Phillipa. One little bit of it went a bit fast for me, and I wanted to be clear. At one point, you compared nucleosome occupancy, those graphs, and let's see, DNA's hypersensitivity and CTCFs. And there was meant to be a difference. Was the difference that the methylation didn't change around those places, but it did with the nucleosome occupancy?
You're talking about these graphs?
Yeah, so tell me the difference. Is it the methylation line, the sort of, that guy there?
Yeah, so this is quite a complicated graph, and I did skip over it. What was your question again? Sorry.
Well, highlight the differences that we're supposed to be noticing.
Right, okay, so if we look at regions of the genome that do have a nucleosome depleted region, we see this dip in occupancy here in the green line, and we also see the dip in DNA methylation as we would expect. This is what we see at promoters, and obviously I've shown you that we can see that enhancer regions. What was interesting to us was when we look at regions that have no nucleosome depleted region, according to our cutoff, we still see this organisation of the nucleosomes, but no great dip here at the centre of the feature, which would indicate that there's no nucleosome depleted region, which is part of our criteria. But when we use DNase 1 data, which was publicly available, and do the same kind of analyses, we see, again, we can detect the nucleosome depleted regions here and the phasing when there is open chromatin. But when we lose the ability to detect open chromatin, we don't see the phasing that we saw with our GNOME-seq technique. Yeah.
Thanks.
Thank you, Phillipa.
No problem.